Good morning, everyone, here at the Biennale in the Italian Pavilion, and good morning to all out there in the world. Today's panel discussion focuses on synthetic biology for carbon-positive architecture from now to 2100. In this context, we can acquire what do 1200 years old Venezia and the centuries much younger Miami Square Islands both have in common, other than their big differences in age. On a geological timescale, both are sinking. Both are exposed to be swallowed by sea level rise, storm surge, swallowed by and burned by heat waves, experiencing environmental, social, cultural, economical, and technological disruptions, bettered by pandemics and future large-scale population displacements. Both urgently need new forms of circular metabolism, bio-inspired innovative planning strategies, incremental resilient solutions for their dynamically changing aqueous environments, new self-adaptive, maybe self-repairing, even growing fluid blue-green infrastructures with a full range of successive living building properties and city systems. Today I will be the moderator who asks the questions. But also expect that the panelists ask questions as well, or disagree or agree. I'm Thomas Spiegelhalter, architect, engineer, and town planner for 32 years, working, building, and researching in Europe, Africa, Asia, and the Americas. And who knows where I'm going next. I'm currently a professor for sustainable practices and resilience at the Florida International University Architecture College in Miami. I'm a Miami research principal for the 3-year EU Belmont sponsored Climate Resilient Urban Nexus Choices Research, addressing the food, water, and energy nexus sectors. For all those here in the pavilion, you see an endless loop video with animations in the background running. It shows the work of over 100 master students designing scenarios, imaginations, ideas, and dreams about Miami's greater islands will look like in 2100. And they and their kids will be the ones who will experience the time towards 2100. Of course, like my students, I also have been immersed and fascinated since my childhood, like many of you, with natural processes and how powerful the metabolic diversity of flora and fauna grows and disappears, dies, and adapts, recycles, transforms. How networks of unknown languages like root systems and fungi surround us. Certainly, when I was a student, like many others, we all had to digest again and again Frei Otto's 1,000 pages and models of research on nature's membranes and cell structures. But today, we are further on the verge of another scientific and ethical understanding, maybe a more significant revolution that opens new ways of genetically writing and coding biology with integrated AI data and sensor-driven nanotechnology on multiple scales. Perhaps we can soon grow natural objects, buildings, infrastructures, and even cities, and not just 3D printed plant-based food, bridges, and objects. With these eminently pressing and lifelong fascinating inquiries, all three invited panelists today are precisely selected experts of influence in a disruptive field of innovations for the future. Please let me first welcome on the panel Dr. Andrew Hassell from San Francisco, California. Actually, from Topanga Canyon at the moment. It's 2 a.m. for him. Andrew will discuss with us his world of synthetic biology, where he's moving from developing tools allowing us to modify genetic codes to tools allowing us to write entire genomes. His research has been critically discussed and shared in hundreds of conferences, TV and TED shows worldwide. of how we are entering an area allowing us to program cells from scratch, how important it is that we are getting involved. Andrew was a distinguished research scientist at Autodesk in their life science group until 2018, which developed software for engineering molecular and living systems. Andrew is also the CEO of Human Genomics, which develops engineered nanoparticles for applications that include therapeutics, vaccines, and gene therapies. In addition, he's on the leadership team of GB Right, the international effort to design, build, and test like the human genome in cell culture. There's so much more to mention about Andrew, but let's do this offline after the panel discussion at our own pace. The second panelist is Dr. Mitchell Joachim. He received his PhD at MIT, and he's the co-founder of TerreForm 1, and he's a professor of practice at NYU in New York City. He also is a frequent project award-winning architect and speaker by dozens of global conferences, TED shows. He was selected by Rolling Stone for the 100 people who are changing America. Most memorable for me was Mitchell's official TED 2010 conference 11 years ago, when he presented his vision for organic architecture grown from plants, including meat production. Mitchell asked, why grow homes? Because we can. Right now, America is in an abiding state of trauma, and there's a cause for that. We have got Mac people, Mac cars, and Mac houses. As an architect, I have to confront something like this. Mitchell won many honors, to name a few: Lafarge Joachim Acknowledgement Award, both Arrow Foundation grant, Time Magazine's Best Invention with MIT Smart Cities. In addition, he co-authored 4 books, among them Supercells, Building with Biology, of which topics he will critically share with us today. Then we have today Dr. Rachel Armstrong, Professor for Experimental Architecture and Living Systems at the School of Architecture, Planning and Landscape, Newcastle University in the UK, and currently she is visiting professor at KU in Leuven in Belgium. Rachel is exploring and experimenting with the very stuff of life. She celebrates the strangeness of the world, even though we have rendered it ordinary, asking how we may design and build our world differently. She establishes a portfolio for design where built constructions are wholly regenerative and live in the environment with their footprints and are synonymous with an aspect of nature carved out by their inhabitants. The architectures proposed within this space are grown from the bottom up and applied to develop next generation sustainable prototypes and systems. Rachel's oeuvre is exemplified in the H2020 FET Open Living Architecture Project and the Active Living Architecture Project, the Active Living Infrastructure Control Environment called ALICE Project, which is an EU-funded innovation award for which she was the coordinator. Her current project scale these principles to urban dimensions, and she hopes to share some of them with you today. Now let's start with Andrew Hassell with the first panel question. Andrew, you hear me well, right?
I hear you perfectly.
8 years ago, in 2013, you said in an interview that we don't live in nature anymore. We put boxes around us. But now we can actually engineer nature to sustain our needs. All we have to do is design the code, and it will self-create. Our visions today, if we can encapsulate them in a seed, will grow to actually fulfill that vision. Andrew, please explain how we can code, script, or write life with growing architecture.
Don't putting me on the spot. I have been fascinated with life, and I study it at the level of a single cell most of the time. My background was microbiology and genetics, but what I find so fascinating is that by writing genetic code, we can program the cell for its life functions, to produce materials that we need, whether it's a medicine, a food, a structural material, to grow and develop into a form, which I think is wonderful. And ultimately, the thing that really captivated me, to duplicate and grow and divide. So one cell becoming two, and so on exponentially. But I have to admit, my view since 2013 has changed slightly. I have become, no, actually even radically, Thomas, because now I believe that nature is so complex and so misunderstood, and it will take us such a long time to appreciate not just the machinery of life, but the relationships between the living systems and the ecosystem. They're so complex that I still stand by the statement that we don't live in nature anymore. In fact, we're the only organism that really likes to isolate itself from the natural world. But I don't think now, I think we have to develop a new relationship with nature, that our ability to engineer living systems will help sustain us in our new isolation from nature, bring nature into it under new terms. But I've become more aware that the engineered cells that we create should be firewalled from the natural systems that we don't yet understand. This happens naturally in the pharmaceutical world. We have bioreactors and containers. It happens naturally in our labs. But I've become much more aware that the things that we're engineering, that all inspired by nature, rooted in the same systems as nature, really should be isolated from nature until we have a much better understanding of the natural world.
Thanks for Andrew. Now let's get to the second round of questions for Mitchell. Mitchell, can you please respond to Andrew's outline with your experience on working with, as you called it before, you published in your book, supercells, building with biology? What do you mean exactly what that is?
Yeah, first of all, super great to be here. And I appreciate, actually, even this topic is something that is still rather rare in this field, at least the field of architecture. And actually, in many other fields, we really don't discuss using nature necessarily in a way that is so intent on creating functions that make sense directly for people in architecture. It's 5 a.m. here, so this is also new for me. I'm not sure I'm even awake. So, but I do this so often that even hearing Andrew's question, or answer to your question, Thomas, I just it seems like it's a second nature for me. But assuming that I'm not dreaming and I'm actually here, and to have a more dynamic answer than I usually have, I will agree with Andrew about the difference between the artificial and the natural, and maybe creating that firewall and the separation between the two. Building with Biology, the book that I co-wrote with Nina Tandon, who is a cell biologist, and she has her own company called Epibone. She's kind of like a Dr. Frankenstein, because the work that she was doing and the work that we were looking at was, can we, remake living parts, in this case tissue, in this case skeletal cells, exactly like they are in the human body, and replace parts of your human body with things that we design, grow, and build in a lab. Certainly a lot of that is geometrically shaping them and then replacing them, such as your jaw after some kind of an accident, in the human body surgically. And the book was written some time ago. I think it was 2014. And even then, what became, the possible is still not something that is practiced in every day, and it still has a long road to go before this is something that is super viable, and certainly the testing behind it. But there seems to be no major stops, and I don't necessarily have this, what we call the precautionary principle, which I do have it in science, but I don't think that there seems to be any reason for us to discontinue this line of thinking. So I understand that we don't understand nature. I understand that we don't understand nature, but there's not a good enough reason for us to put on the brakes right now. So supercells, the book that you were referring to, is very much a kind of a clarion call for opening up this beautiful kind of world or cornucopia of possibilities in design, in architecture, in medicine, and in an enormous amount of other fields. Biotech itself is on the premise that everything is made from cells on this planet. So we can continue to do that. We'll just choose more specifically the designs that those cells produce. So I'll put a pause there at my 5 in the morning, and that probably is the most unfit I've ever given.
Thank you, Mitchell. We will get back to you and also to Andrew with more specific questions, but let's go to Rachel. Rachel, you hear me?
Yes, I do, and I'm so sorry. My video has gone totally blank. So the only way I can appear is as a little virtual icon. So actually, my software seems to be in a place at 5 a.m. itself. So it's really refusing to cooperate with me. So I'm impressed by Mitchell's eloquence at such a time. My software is just having early morning teething problems.
So let's get to the question. So, Rachel, what are the biological architectural systems or protocell technologies that you have used or you will be using in aqueous environments of sinking Venice structures? Could you explain more about this?
Yeah, well, since I can't appear, if I can just throw up some slides and I'll race through them. I'm not going to give a big presentation. Please. Okay, great. If I do this, then at least you've got something to look at other than a tiny icon. So I think this question about synthetic biology is really critical to these times. If we're going to change building performance, then we need it to at least, at the very minimum, be aligned with nature, not fighting it. And the only way we're going to do that is by working, as Mitchell and Andrew have said, with the actual building blocks of nature itself. So I started trying to figure out how to do this around about 2008. And I wanted to figure out how to work with metabolism. So before it comes to biology, which was always irreducibly complex, could I strip something down so that the flow, the stuff of life, the things that cells do inside, was as it's possible simplest? And could I think about how that might actually work, first of all, and then how we could design with it in a natural way? And of course, the great place, which is sitting in a petri dish, which we call a lagoon, is Venice. So Venice is a city that already has its kind of cellular medium around it. And so the idea was to place these very simple protocells that literally mineralized the salts in solution in the lagoon into a kind of kettle scale, a very primitive reef-like structure around the foundations of the city, because as the Grand Navi come through the or past the Grand Canal, they suck out the water from under the city, exposing the wooden foundations and causing them to rot because the bacteria love that. But when I started to do this work, we saw that actually natural organisms, so algae, calcium carbonate making algae and bacteria, were already making these little thrombolites, like little nature-made stones around the sides of Venice. So the idea was then that Maybe there was a collaboration between the protocells and the natural systems that would respond to the rising waters and create this protective layer around the city. So these are some visualizations kind of going from this, you know, something that you can just about see with your naked eye to something that was an urban scale. But the question, and that gives us ways of thinking about, you know, design differently. There's this stuff excited me so much that I went and wrote a fiction book, but it enabled me to think beyond the traditions of what we would call buildable architecture. So in this stage, this is like a parallel reality of Venice, where because of this dynamic masonry, we squeeze out architectural structures, a bit like Nina Tandon's work, really, using the techniques of plastic surgery and dental tools in order to kind of push materials into spaces. So once you've got these kind of squidgy infrastructures, we could start to think about building buildings differently. But the interesting thing for me is, you know, round about the turn of the millennium, we started to develop molecular biology tools that didn't just allow us to see individual cells, but populations. And that has created a revolution in our thinking about the microbial realm. So one, this human microbiome, we knew that we were in a world of bacteria. We didn't know just how much they are part of us. And so this, our own bacteria inside our guts and on our skins and just everywhere in our bodies, forms a kind of necessary organ. You know, you think about how sick you get when you have antibiotics and you kill your natural organisms, and then stuff comes in from the outside and you have tummy problems for ages. And so our relationship with our microbial cells, almost like this a residential organ that helps us produce skin softening agents and helps our mood and helps us digest our food. That's really weird, and that's really great because it's starting to challenge notions of these discrete identities. The other thing is that as we are full of bacteria, of course, we spread them everywhere. And so our cities too, the urban microbiome and the microbiome of the built environment were kind of interrogated and discovered and mapped by people like Jessica Green, who's also another TED fellow, looking at, you know, the microbial landscapes all around us using these tools. So the thing is that these tools are actually all around us, but we're not really doing anything like design with them. So synthetic biology does give us that opportunity to think about what it means to design with these invisible realms. And in fact, in 2011, Philips Microbial Home was a proposal where the lighting company was looking at, can we have a perfectly circular kitchen where all the kitchen waste actually becomes degraded by microbes, like a kind of designer soil, and then reutilized within a kitchen environment? Well, that was theory in 2011, but in 2016, I put together a team of fantastic people to develop this multi-bioprocessor system called the living architecture. So there are three, I'll show you the types of bioprocessor, but the idea here is actually to design with populations of microbes. So trying to take the microbial to the human scale, and instead of let's say, to try and think about resource circularity, so looking at it as a regenerative thing. So it's more like a multi-utilities domestic system than it is a system of material production. That doesn't mean we couldn't program it like that, but this was really to think about how we might get rid of the notion of waste and wean ourselves off fossil fuels. So we thought about a circular resource flow system for the household. So you can see there are three major forms of bioprocessor here. This is the kind of stuff they do. They make electricity, they remove waste, they neutralize pollution, they can reclaim phosphate from detergents. And some of that stuff can go back into the household to support our activities of daily living. And some of it can be put into the environment just as literally part of the atmosphere and nature. And these are the three little homes for microbes that we made. So we think of those bioprocessors as being little designer cities. And what's cool about microbes is that environment means everything to them. So in other words, designing environments helps us create a selection pressure to the kinds of microbes that we invite into these spaces, particularly with this microbial fuel cell, which is a battery system, an organic battery system discovered in 1911 by somebody at Newcastle University called Michael Cress-Potter. Essentially here, an anaerobic biofilm produces electricity, low power electricity that can then be used to do weird things like drive artificial intelligences and lights and all kinds of stuff that this complex system needs. We've got the algae photobioreactor, tiny trees and cows that are metabolizing carbon dioxide and light. And then this is Andrew's world, the synthetic biology and the synthetic bioprocessor at the end, where we've literally got two stories here. We can think of this house as being organized in two stories. On the top level, we have engineered microbes that overproduce sugar. So this is Charlie and the Chocolate Factory. They make so much sugar from light that that sugar diffuses through the first floor level and down into the basement where it's dark. And we have two microbial species that don't normally talk to each other and hang out as a consortium. In other words, that's kind of like a, like a little community. And these two guys here, we know that they're very well characterized. So that means they give us a whole bunch of tools that we can use to create new metabolisms. And this is where we can detoxify nitrous gases, we can reclaim phosphate, and they can do this because we're feeding them sugar. So these guys are on a sugar trip, and now we'll do anything we ask of them. So that's how that works, complete organic bribery. So from that then, we have an integrated system. We can make lots of little chambers that sit together and, they communicate through these biomolecules and process our waste into stuff that we can use. And you can see we can start to design with this at scales, at household scale. Our urine and wastewater goes into a settlement tank, and we can think of those bioprocesses as little stomachs, and this is the poopy bit, and everything's really safe. And you can see here that we've got the genetically modified organisms here. I just want to talk about that for a second, because the way we use them is really, it's not to say that we want a genetically modified system in the home. What we're doing is actually cheating with biology to see what biology could do if we found natural organisms. And we know that literally microbes now are like the top of an iceberg. We only know a very few of the potential species. So J. Craig Venter went out to the Sargasso Sea, went fishing, comes back with about 300 new species of microbes. I mean, it's on that level of order. So the idea is it possible for these microbes to do these forms of work and then to build a natural library so that, 5-10 years in the future, after we've all been fishing for microbes, we actually get our little a la carte natural microbes to perform these extra little functions. And the thing with this is, I think that in your title, you were talking about carbon capture and synthetic biology. And it's very true that most of the life on the planet we know uses carbon as some kind of fuel. And the reason they use that is because they use it to pass electrons around. And I'm really interested in this electron part. So, call it the bioorganic aspects of synthetic biology and their ability to move electrons around through their weird and wonderful metabolisms. And this is what I call a post-human home. This is only inhabited by microbes. It's a collaboration with Cecile B. Evans. So we've got all the little microbial stomachs there on the left. The ghosts from the past, present, and future in her digital work are on display here. And so this apartment is only occupied by these microbes. It's fed once a week in this black box here. This is its guts, all that produces the electricity. And here's the little poopy bit. So that is cleaned once a week. And there's no need for any human occupation. Obviously, some of us might want to live in such an apparatus, but this was really trying to embody the idea of, what can we do with, kind of microbial exchanges at the level of human scale. And it really starts to help us see that, in order for these kinds of processes to be real, to inhabit domestic space, we need different infrastructures. We need to be able to feed and think about the kinds of support systems that are needed. So we're really thinking in terms of how, we don't just think about a house, we've got to think about how a bioregion supports itself, not just very locally. And I know I'm going on, but there's a really interesting bit, which is when the pandemic came, we'd really not thought about, so there's the microbiome, and we had these, we had city councils around the world trying to monitor prevalence of the virus by going down into the sewer and, having a look at how much COVID there was, knocking around that nobody was telling us about. But what was interesting is that the Living Architecture project is taking urine and fluids from our bodies and could deal with human waste. So we could use that actually as a sampling, not just a sampling system, but when we look at the microbial fuel cell, which is a little city of microbes called a biofilm, they're self-regulating. And in 2016, 2017, when we were trying to deal with the biosafety issues of the Living Architecture project, we thought of the microbial fuel cell as an immune system that could catch genetically modified organisms that might have crept out of one of the bioprocesses. So there was a specific experiment that looked at whether the microbial fuel cell could capture a pathogen like hepatitis B. And as you can see, when you close the circuit and you recirculate the wastewater through the microbial fuel cell, the pathogen count goes down to 0. So this is really very exciting, because actually now our buildings could potentially be first-line immune systems like they are in our bodies. And so this one is really about how do we communicate them. This is the ALICE project, using, because once we start to excrete electrons, that is both power for a digital system and it's a form of data. And we can start to use that to build different kinds of relationships. And of course, we can start to scale them to the sides of gardens. These are real things. We can use them to power. This is at Glastonbury. They've had this P-Power for 5 years now, running with Oxfam. And so party goers can charge their phones, the screens are lit, and they can play computer games just because they put their P to good use, and that's all courtesy of microbes. And so there are now, networks of scientists working around the European Union trying to develop these bioelectrical systems, which will help us detoxify brownfield sites, and we'll put big membranes in them. As I said, this is an apparatus that grows marijuana from the P of Guys in Amsterdam, very Dutch. And then this one is thinking about how we might actually use those combined utility systems as Bill Gates has envisaged, using the microbial fuel cells to have refugee communities of up to about 30,000 people, literally having clean water, sanitation, and a small amount of electricity that keeps people safe at night. But it doesn't just have to be an off-grid environment that's very basic. If we can rethink our lifestyles through 12-volt systems, which could be usefully charged by these organic systems, then I think all kinds of interesting possibilities become reasonable. And this is where I think that the kinds of visions that Mitchell and Andrew have been proposing, really then start to come into their own. So I did mean to keep that short, but since I can't appear as a face, you got my slides instead.
Thank you, Rachel. Excellent, fantastic imaginations and applications already in place and will be incrementally more applied to cities and infrastructures. That brings me to a question which makes me deviate from my script. And this is spontaneous. I'm becoming in a couple of days grandpa. And I'm working for years on a research. It's based on Wilhelm Flusser. You might know him, the philosopher Wilhelm Flusser. And he was looking at octopuses, and it's asking the question, why 3.6 billion years ago the octopus developed in a liquid space, whereas humans developed to go upright on 2 feet. And the impact on our world is the industrial Cartesian space. So looking at the fetus, which already develops in a microgravity based space, And gravity loading is necessary for the physiological development during second half of pregnancy and all the phases after that. Once this baby Theo of my daughter Fenna is born, the world will train him to go upright. What if in the future, towards 2000 and beyond, we are not using industrial Cartesian systems anymore, and we are including liquid spaces? So architecture becomes a liquid space. Can anyone kick in, or am I far off? I'm just wondering why the liquid space has no ceilings, has no boundaries like walls. And there's microgravity as well, but it's an unlimited space of opportunities. And if you're amphibious, you have more options.
Can I I'll quickly respond. I know I've spoken a lot just recently, but the author William Bryant Logan says that during the origin of life, we learned to contain our own seas. And I think that's the difference between the octopus and the land-based animals, that our seas are internal. So it's a really good question as to what that relation, why we've chosen specific relationships with water. And I think it's up for grabs. So I'll stop there.
Yeah, I want to ask Andrew, riding life, what do you think? What happens in 2100? It's also an ethical question, of course.
That's really interesting. And by the way, Rachel, I love your presentation. I love the prototypes you're doing. And the thing that really resonates with me today, and I think a lot of this, Thomas, is in the decade that I've been going and really communicating, the power and potential of these technologies is I've had 2 children of my own, and just watching this incredibly complex being formed from a single cell is remarkable. And both my kids are lab grown. They were booted up in labs. They're IVF babies, and I tell them that it's so funny. They'll be playing with other kids in the playground, and they'll say, "Yes, I'm lab grown. My brother was made in a New York lab, and actually in a San Francisco lab. Yeah.
I'm a C-section."
But,
oh God, I can't wait for the time where all our kids try to out-tech each other.
but the universal of life is water. Water is what allows our life to really to activate. And when I was early in my career, I would take bacterial cells and I would remove all the water from them with a freeze dryer, and they'd become a powder. Add water and they reconstitute. Of course, we do the same thing with yeast if we're baking bread. And the yeast is freeze dried. So water is absolutely crucial to this life process. But we live in an open system, and I think we need to isolate ourselves from most nature. If we were just to go swimming in the ocean and call this our new home, it won't be long before some predator comes along and decides to eat us. But the thing that I think is really important for us to appreciate today is that we need this full circle, this full closed loop. Earth as a planet is really a closed system, other than the energy coming in that keeps everything circulating. But we need to start being able to do that on a societal level. It's wonderful if we can do it at the home level, but we have to start doing this as a societal level if we're going to put things back into balance. So for the last couple of years, I've been studying things like the life support systems on the International Space Station, or the amazing Biosphere 2 project that was started in Arizona in the 1990s to create a closed environmental system, or even something like a submarine where a crew will go out on missions for months at a time and have to maintain these systems. This is what's inspiring me now. Can we take the lessons from nature as you've so brilliantly shown, Rachel, and start to incorporate this into holistic systems where we can live inside of them and be sustained, and recycle our waste, produce our foods, generate the electrons that we so need for our everyday life today.
Thank you, Andrew. Mitchell?
Yeah. Rachel said troglodytes from protocells. I picked up on that one. And pea-powered pot in Amsterdam or someplace in the Netherlands, and everything else. It's too much for 5 in the morning. And then you're asking about what if we're liquid? And I think Andrew gave a phenomenal answer. So I almost want to say, good job. But if I had to add to that, I mean, the mind is already liquid, right? And there's many fields that have kind of given us that capacity to think about the separation between our mind and the body, both on an ontological level and one of just pure imagination. So, we are very fluid in how we think. We are amazing in our capacity to think of other phenomenons as us in nature or other natures itself. So I don't, we're simply just not limited. And we have a kind of was it an octopus or a squid? I don't remember which one you were referring to, Thomas.
The entire family.
Yeah, any of these fishy bits, many legs.
Any of these.
Yeah, so it's no problem even for a young child or a sophisticated adult to rethink, and I just mean think conceptually our worlds as if we were another. It doesn't necessarily mean it's useful, but it does eventually develop new modalities and helps us kind of project what a possible future would be like in such a realm. Andrew mentioned the International Space Station. This might be a more specific, if not practical use, where the body itself is no longer viable. And we're in a world that's agravitational, and that everything we've been used to as far as how we design and think about movement and spaces and volume and mass all goes away, and we have to rethink what it means to be human in that universe. There's not enough experimentation there. I think an individual by the name of Zbigniew at the Rensselaer Polytech Institute has been looking at worlds like this. And we're born into a world like that, as Andrew pointed out, from a single cell and a fetus. It's there. And I love Rachel saying that we contain the water within. But we've evolved for other reasons. I would think that even I'd love to run a lab on what it's like to be liquid humans. But the internet and where technology drives us, especially when we're so disembodied all of the time, we are that in many ways. So it's just a matter of taking it to the point where we are literal and physically become liquid. It's a very, very new dimension. But there are moments in life where that you can kind of get that feeling, I guess. Not quite there yet. And that'll be my 5 in the morning answer. I might say, well, when we get a moment, I'll show if that's possible. I think we're limited on time. We've been doing.
Okay.
Well, we've been doing some work.
You can share.
So all right, I'll share a slide.
Please.
Insanely quick. Let's see.
I got green light. We can extend.
Okay. So I don't know if we can see.
Yes.
All right.
Perfect.
You see worms and foam.
Perfect.
Yeah. So a liquid being might be something like this, which is a mealworm. This case, there's an Amazonian mealworm. And these phenomenal creatures have been our client as architects for about 2 years. It's a paper that came out of Stanford University where it looked at how mealworms consume styrofoam. And to me, this is a phenomenal architecture because they are both architects and chefs and also inhabitants of this particular world, which is just made of essentially e-waste. Styrofoam is an awesome and wicked problem that we have in all of our cities, both in the United States and in Europe and China. Can't dispose of it easily. It's very difficult to upcycle. What do we do with it? Well, here, mealworms actually love to eat this material. And they do that naturally with no modification. And they actually turn it into an architecture without consideration of gravity, because like it might as well be liquid, or it's in a transitional state. They eat it in every single direction and in all directions. Sometimes they'll hit each other face to face. Sometimes they'll go up, down, sideways, doesn't matter, but they're consuming it at a very slow pace, but they devour it and they turn into something called frass. And that frass is then excreted from outside of their intestinal systems, their enzymatic systems, and then eaten again until the styrofoam disappears and becomes mulch or garden waste. So we developed these bioreactors where people donate, as if it was a Mayan temple, bits of their styrofoam trash and watch it digested by these little creatures, our client, thousands of mealworms that will absorb your styrofoam and turn it into garden mulch. And then that garden mulch is contributing to the local biodiversity as it adds to more flora and fauna. And then the mealworms themselves become darkling beetles, move throughout the rest of the shelter and get consumed by lizards and birds and other types of creatures in the area. And this is the actual digester. There's a clock on the top that shows the time constraints it takes for different types of materials to be naturally processed in the environment. And some, like styrofoam itself, can take 500,000 to 1 million years. So here, mealworms do that in a matter of weeks. Here's that digester system. In the back is a farm for more mealworms, making tens of thousands of mealworms that are simply grown to consume your styrofoam. They're produced in these little micro farming elements in the back. And then you throw your styrofoam in the front and watch biology at work. And here it's kind of a combination system between a public art installation, architecture, and everyday systems you'd find in nature being put to work for humans. And that is a close-up of the digester system itself. So I just wanted to kind of show that. So I find these guys to be in some level very liquid because their environment that they move in has no sense of direction. And another project we're working on is actually controlling them to develop actual spaces that are about a subtractive architecture. So very similar to places like Cappadocia in Turkey, or where troglodytes, as Rachel pointed out, normally dwell. So this might be a new way of developing troglodyte architecture. For what purpose, I don't know yet, but I'll let you know when we get there.
We get there in 2100 and beyond. Yeah, thank you. Fantastic work, Mitchell. Andrew, you want to kick in with some maybe closing projections to make us hopeful that we can grow fins and other parts. And you know.
By the way,
yeah.
Mitchell, that's absolutely fantastic. I did not know there was any organism that could efficiently digest that material, and you give me hope. That's. And I have to admit, your work with the idea of growing homes is absolute, has always been inspiring to me as well. I think the master regulator for this, Thomas, like these are wonderful examples, and again, nature is so incredible that we have organisms that already exist today that can digest some of our most toxic waste. It reminds me of some of the work by Paul Stamets as well, with his wonderful fungi that could go and remediate some of the materials. When it comes from an engineering perspective, though, I think we are just getting started. Much of my work has been looking at how do we write the genomes of these organisms and thus program them, and it's humbling that in really the 15 years that I've been focused on genome synthesis, we've just crossed the threshold into being able to engineer a complete microbial genome. To my knowledge, there's been 3 microbial genomes built from scratch. So we have a long way to go to be able to program these capabilities. But what gives me hope is that the writing of genetic code, the equipment for doing that, and the language that of the genetic language itself moves at a pace that should be super exponential and move even faster than our historical experience with computers. So even though it's a very slow start, because the genetic language is universal and conserved, we'll get better at programming it very quickly. And because we all need to be able to write and assemble the genetic code to be able to program these organisms to become foods, to digest our waste, to provide energy, etc. That technology is going to move very quickly as more and more engineers, scientists, and creators advance it. So today it is a significant challenge to write a cellular genome, but I'm confident that by the time my children, 3 and 6 years old, are my age, it will be something that is done every day in part of creating and maintaining this world that we live in.
Thank you, Andrew, for giving us heads up for the future. Rachel, do you want to give us some concluding words? A short narrative?
Yes, I will make it short. I think I echo everything that Andrew said, and I think the thing that I would just want to add is that what's very interesting about nature is that unlike how we work with computer codes, which is human driven, so it comes, so from Turing, looking at morphogenesis, thinking about mathematics and how we kind of organize concepts. What's really cool about living cells is that they push back. So as we try to provoke and invent, they will help us or refute us. So I think this kind of distributed agency in addressing challenges, I won't say solving, because the second you solve nature is death. So things keep moving, things are always on the move. And so I think what's very interesting about this age of design and engineering is that we now have subjects that push back, and that for me gets really exciting.
Thank you, Rachel. So we are at the end. I just want to thank you all for participating, and I hope we could energize the audience with this emerging and really high-level research and applications. Thank you all, and especially I want to thank also the Italian Pavilion team, Benetta, Danielle, and Gianluigi. Thank you all, and please have a wonderful day and stay safe. Thank you all. Bye-bye.
Wonderful. Thank you.
Thank you. Bye-bye.
Bye-bye.